The third line of defense in the human body is the adaptive or acquired immunity. So either one of those words are interchangeable. And this is often called our immune response or immunity and consists mainly of lymphocytes and serum proteins called antibodies. We have these protective mechanisms of external barriers, which we talked about previously with the first line and second line. Second line being the inflammatory response. Now we're going to be discussing the third line, which is adaptive immunity. Inflammation is the first responder that contains the initial injury and slows the spread of infection, whereas adaptive immunity is that secondary responder. And that augments that initial defense against the infection and provides that long-term security against reinfection. Innate immunity, especially inflammation, and adaptive immunity are highly interactive and complementary. Remember how I said that these things are going to overlap? But we do have to talk about it in a stepwise process. So hopefully once you go through all of the immunity lectures, it will all make sense. So we talked about the innate response and the players that came in here, macrophages, dendritic cells, neutrophils mainly. Now we're going to be coming over here to the adaptive response, where we're really talking about B cells and T cells, antigen presenting cells, and things that are involved with that initiation of the adaptive response. Inflammation and adaptive immunity differ in several key ways. First, adaptive immunity is much slower, all right? So it is more specific, but much slower response. It takes a while for it to get up and running. The components of inflammation pre-exist in the blood and tissues are activated immediately. Adaptive immunity is inducible, meaning it has to be acted on by an agent. So it has to be induced. And I have a slide coming up. Meaning that the lymphocytes and antibodies do not pre-exist and must be produced in response to that infection. Another thing is the inflammatory response is similar for whatever insult, regardless of the differences of the cause of tissue damage, or whether it's a sterile or contaminated. The same happens with the inflammatory response. The adaptive immune response is exquisitely specific. The lymphocytes and antibodies induce a response to a particular infectious agent and are extremely specific to that agent. And that's how we get memory. So when we talk about inducible stimuli, must be elicited, the response does just not exist. So again, exquisitely specific and highly specialized. We are also able to recognize self from non-self, and we're going to talk about autoimmunity later. There are multiple ways that we induce stimuli. Infection is the most common: viruses, mycobacteria, parasites, and fungi. Other ways that we will elicit is that autoimmune response, where the body turns on itself, potentially foreign material, and then some cancers as well. So the collaborative beneficial nature of inflammation and adaptive immunity can on occasion fail, and we're going to talk about aberration in both the inflammatory and immune responses, such as allergies, autoimmune disease. Those are the big ones. So again, a better slide to look at the players in adapt immunity. So we will have these antigen presenting cells. That's what APC will mean moving forward, is antigen presenting. We have T cells, and they are T helper cells, or CD4 plus, or we have CD8 cells, and those are the cytotoxic. Then we have B cells, and they will either go into memory cells, or we will get antibody FAC, which are called plasma cells. And we have macrophages, dendritic cells, and they have a lot to do with that antigen presenting. Okay, so the main cells in the adaptive immunity are the lymphocytes. And now when we take a look at that progenitor hematopoietic stem cell, we are now going over to this line over here. Multipotential hematopoietic stem cell. We then differentiate into the common lymphoid progenitor. We either get natural killer cells or we get a small lymphocyte. The small lymphocyte will further differentiate into either a T cell or a B cell, and then we'll get further differentiation, which we're going to talk about in a few minutes. They are present in circulation and also the lymphoid organs, such as the spleen and the lymph nodes. And then we also have this situation where we get clonal diversity, and then we have clonal selection. So we're going to talk about those two things coming up here. So they develop from precursors in the primary lymphoid tissues. So for the T cells, that's going to be the thymus. And then for the B cells, it's going to be the bone marrow. And again, just remember the T and B make sense because of where they're coming from. So we get kind of two phases of developing these receptors and clonal diversity, and this actually happens in the fetus. So before we are exposed to any antigen, it is encoded in DNA sequences that we has these receptors that are going to recognize antigen. That happens from the DNA. So before it's ever exposed to any antigen, we are able to recognize antigen. Hundreds of millions of antigens. It's actually 10 to the 8th is what they believe are circulating as immunocompetent lymphocytes. Each B and T cell and its progeny express a single antigen receptor. On a B cell, we will have a receptor. On a T cell, we will have a receptor. So the lymphocytes leave the primary lymphoid organs as immunocompetent but naive. So the fetus is where clonal diversity begins, and that is antigen receptor development. They are considered naive because they have not been exposed to antigen yet. So here we have the bone marrow, we have that lymphoid stem cell, then we differentiate into B cells and T cells. T cells migrate to the thymus. B cells stay in a special area of the bone marrow for their clonal diversity. This is what we consider once we get out of the primary tissue, the thymus or the bone marrow. Then we go into circulation and then migrate to secondary lymphoid organs such as the spleen or lymph nodes. So here we have naive T cells and naive B cells, meaning they can recognize antigen, but they have not been exposed to antigen. So then what happens is they kind of hang out in the spleen and the lymph nodes, and then they are waiting for antigen. So clonal selection comes in when antigen exposure happens. What happens then is we get multiple types of T cells, and we get 2 types of B cells. This is called clonal selection. So this is proliferation and differentiation of the individual B and T cells with those 1 receptors. So let's say we have a little bacteria that we get exposed to, that T cell or B cell receptor, it comes in here, that epitope recognizes it. Now we are going to clone those B and T cells because we have that activation. So we will get regulatory T cells, cytotoxic T cells, memory T cells. We get T helper cells. The B cells, this is humoral immunity. We're going to get B cell memory. We're also going to get antibody factories called plasma cells. This is ultimately what gives us our memory and the ability to fight off infections again and again because we elicit this response. First phase, clonal diversity, which happens fetal tissue in the primary lymphoid tissue. Secondary is clonal selection, which that is antigen exposure. This is where antigen presenting cells come into play. So how do we get that exposure? The dendritic cells and the macrophages will break off and phagocytose, and then a little piece of the antigen will be populated. The B and T cells then get exposed and they link up. Much more to do on that coming up. So here is what that looks like with clonal selection. So here is a B cell. This is basically the immunoglobulin. We have parts of this called the heavy chain. This is the FC region. And then we have the light chain, which is the top portion. Here is that epitope. So that is what is going to recognize antigen. This little thing here, this is what happens with the DNA selection and diversity in that first phase. Then we have antigen that comes in. Then we're going to get clonal selection, and that gives us the ability to elicit a response. Each receptor created through epitope. On the B cells, we call them antibodies. On the T cells, we call them receptors. So that's a little variation to understand that they are called different things related to B cells and T cells. So let's take a look at the B and T cells a little bit closer. The T cells, progenitor T cells, bone marrow, migrate to and develop in the thymus. TC receptor undergoes rearrangement. That is that epitope. Progenitor cells will then evolve into either CD4 T helper cells or CD8 cytotoxic T cells. Those are the main ones, and those are the big ones that are in the response. We do have some other T cells, and they are more regulators. So we have T regulator cell, and this has a lot to do with self-tolerance. We have T helper 17, again involved with extracellular pathogens and autoimmunity. We have T helper 22, and that has a lot to do with wound healing. All right, so they will differentiate. You have your naive T cell, no antigen when it's naive, but when it gets exposed to antigen, then it will differentiate into any type of these T cells. The ones that we talk about the most are T helper, and then also the CD8 cytotoxic T cells. They regulate the immune response by helping the clonal selection process, so those are the T helper cells, or suppress and limit the immune response, which is the T regulatory cells. Both B and T cells differentiate into long-lived memory cells as well, and they exist for decades and sometimes the life of the individual. They use the TC receptors, T cell receptors, as surveillance for antigens, and then also recognizing major histocompatibility complexes. That's what that stands for. And this is how we antigen present. They recognize antigens on the surface of the macrophages or dendritic cells, such as virally infected cells, tumors, foreign bodies, release of cytotoxins, and also involved in delayed hypersensitivity reaction. So CD4 plus activation. So we have these major histocompatibility complexes. And what you need to remember, if you can't remember, CD4 goes with MHC class 2. Just remember the rule of 8. 4 times 2 equals 8, if you can't remember. So CD4 activation is related to extracellular antigen or a foreign protein. So here we have a macrophage or a dendritic cell or macrophage. We get antigen, we get phagocytosis, we get that ligophagosome, we get it broken down, and then what happens is we have this presentation of it. So class 2 histocompatibility molecule is this little guy here. So it basically takes it to the outside of the dendritic or macrophage, whichever antigen presenting cell it is, and it puts it on its cell surface. And what happens then is that we have T cell receptor for that antigen. Remember, highly specific. This T cell was already having that clonal diversity from fetal and primary lymphoid, already able to recognize it. It presents it on the macrophage, and then it basically links up. So class 2 is related to an extracellular antigen, phagocytosed, processed, and then presented as MHC class 2 histocompatibility molecule. So that's what this is here. It is expressed by antigen-presenting cells. B7 binds with APC CD28, providing second activating signal, and we ultimately get that presentation. So then the activated CD4 secretes cytokines that help the inflammatory response. All CD stands for is cluster of differentiation. So we just call it CD4, CD8, whichever one we're talking about. So these major histocompatibility complexes are glycoproteins found on the surface of all human cells except red blood cells. So now when we talk about CD8 plus activation, so CD8, 8 times 1 equals 8, and that's an MHC class 1 complex. So here is a dendritic cell. Here you can see MHC class 1 over here, and then we get activation of CD8 cell. So this is going to be a cytotoxic T cell activation if we have CD8 plus, okay? So CD8s are the cytotoxic T cells, and that basically means cell killing. So this is an intracellular antigen from cytoplasm proteins that are processed. So something is going on within the cell. It is not related to extracellular antigen processing. What happens is that all cells except red blood cells express this major histocompatibility class 1 receptor. What happens is something happens within the cell that will then have the cell say, we got a problem going on here. Maybe there's a virus in there, or maybe there is cancer. And the cell basically is going to activate apoptosis. The way that it's going to do that is bring in the cytotoxic T cell. So it's going to present whatever is going on, and then the CD8 cell is going to bind to that epitope, and we're going to get activation. So it's considered an endogenous exposure. So cells infected with a virus or cells that have become malignant, the antigen-driven signal for the T cell clonal selection is recognized by that T cell receptor combined with CD8 binding on that MHC class 1 molecule. There are a lot more steps, okay? Like I told you, we're not going to, this is not a degree in immunology. There are a lot more steps going in here. What you must understand is just how we get activated and what happens when we get activated. Interleukin-2 provides second signal. T cells are activated for killing. We get secretion of perforin and granzyme, which is going to then get apoptosis of the cell. We also have this FASL, which binds on target cells and also activates apoptosis. So we actually get 2 ways that we are going to activate apoptosis. The other thing that happens here is we have CD4 T regulatory cell activation. So you can see this here. We have CD8 T cell. We also have CD4 T regulatory cell. That is going to in and helps get rid of this damaged cell. We call this co-stimulatory adhesion molecules. Also, we're going to get some cytokines that are necessary for the proliferation and maturation of the C cytotoxic T cells. What you see here is that the CD4 cell comes in. We have CD28 receptor, CTLA4 receptor. We have interleukin-10, goes on to the interleukin-10 receptor, so that's that cytokine. So then it's going to go over here and then also activate some other cytokines that are going to go over here and prime the CD8 T cell so that it comes over and does its job. You don't want these things not to have a second fail. So we don't want just one way to activate this, because we could get a lot of cytotoxic T cell activation, right? If we just had major histocompatibility class 1 activation without the CD4 T cell coming in and saying, yes, okay, this guy's bad. We have secondary co-stimulatory adhesion molecule activation. Without this second fail-safe, we would have a lot of killing going on. So this is a secondary measure to protect us from basically killing our cells that did not meet criteria. So we have to have that secondary co-stimulatory molecule with the cytokine release to have this all happen. So those cytotoxic T cells require cytokines, interleukin-2, to become activated in response to antigen presenting. The other thing that happens with this is then we will get some T memory cells. And that is related to these interleukin cytokine receptors and develop in that population of T memory cells, and they have the capacity to rapidly respond to further exposure of the same antigen. When we get the cytotoxic T cell activation, this is what we call cellular immunity. Cell-mediated cellular immunity, and remember we have 2 types, cellular cytotoxic or humoral body fluids. And cellular cytotoxic is T cells and humoral is B cells. Okay, next up is the B lymphocytes. So these are immature B cells produced in the bone marrow. They still remain in the bone marrow for differentiation and undergo immunoglobulin arrangement, clonal diversity. They become the naive B cells. We have those B cell receptors, and they will express IgM and IgD. So the naive B cells or immunocompetent B cells, they will encounter an antigen for the first time. Only those cells with specific B cell receptors complementary to that antigen's determinant sites are stimulated. So we have to have a specific B cell receptor for this specific antigen. This antigen will not be recognized by any other B cell receptor. Another B cell will recognize a different antigen, 1-to-1. So what happens then is they proliferate and differentiate, that clonal selection. And they're either going to go into a plasma cell or they're going to go into memory cells. When we talk about the plasma cells, these are your antibody factories. Memory B cells are going to live long term and provide memory. So each plasma cell is a factory. So we get antigen recognition on a single B cell receptor. This is going to differentiate into that specific antibody producer and that specific memory cell. Again, we have interplay with major histocompatibility complexes, CD4 T helper cells, and it basically is that secondary assistance to make sure that we don't set off an immune response without having all aspects of the immune response working together to make sure that it's activated appropriately. So the B cells differentiate into long-lived memory cells. They remain inactive until subsequent exposure. So then they are inactive. Upon re-exposure, the memory cells do not require any further differentiation and then will differentiate rapidly into new plasma cells. So really cool. So here is what it looks like with B cell activation and antibody production. So we have that antigen recognition. So we have naive immunocompetent B cell with IgM and IgD on it. It gets activated and we have that helper T cells and other stimuli. So that's that CD4 T helper cell. And then we're going to get proliferation of the activated B cell. So they are going to be clones with that activated recognition of antigen. Then we are going to have differentiation. We will get antibody secreting plasma cells. We will get IgG expressed B cells. And then we're going to have the IgB cell differentiate into memory B cells. We have antibodies. We have 5 different antibodies. And what happens is we have this isotope switching. So a major component of the maturation is this class switching. So this is a process that results in a change in the antibody production from one class to another. And we have 5 types. So the antibodies are gamed is the best way to remember it. So we have IgG, IgA, IgE, IgD, IgM. There is that heavy and light chain. We have antigen binding site. We have this variable, and this is what ends up class switching. So the immunocompetent B cell produces IgM and IgD before clonal selection. Encountering antigen, the B cell has the option of changing the class of antibody secreted to form one of the 4 IgG subclasses, or one of the 2 IgA subclasses, or IgE. Or it can continue to produce IgM. This is called isotope switching. Then IgA, IgE, and IgD are also produced. So IgA is a dominant secretory immunoglobulin, and it's to prevent the attachment of pathogens to mucosal surfaces, such as GI tract, pulmonary, genitourinary tracts. IgE is a special class of antibody, and it's designed to help protect the individual from infection with large parasitic worms, such as helminths. It also can be activated in common allergies, and this is huge for our allergic responses. So parasites usually invade mucosal tissues, and in a response to those parasitic antigens, the variety of antibody produces that class switching to IgE secreting plasma cells. So that's when IgE would be class switched. Mast cells have that affinity for IgE, and mast cells, remember, are big in the inflammatory response. IgD, so this one has unknown functions. They're not really sure exactly. May bind antigen on B cell surfaces, and there's a small amount in the serum. So when we look at our functions of antibodies, here is a synopsis. So protect against infection. We have direct effects, which are neutralizing antigen by blocking or binding the actual antigen. Second thing that they do by direct effect is agglutination. And that is defined as clumping of insoluble particles that are in a suspension. Precipitation is making the soluble antigen into an insoluble precipitate. Basically what happens with antibodies then is they protect against infection by covering the sites on the microorganism that are needed for attachment. So that's what this is. So we have those specific antibody receptors, and they're going to bind So let's say this is a respiratory tract cell. This is the antigen. Antibody is going to do is block the ability of that antigen to attach itself. That is neutralization. When we give attenuated or weakened vaccines, it's going to induce this neutralizing antibody production. So then other things that happen are bacteria secrete toxins that harm individuals. So bacterial toxins, such as with tetanus or diphtheria, they bind to surface molecules on cells and damage the actual cell. And then it's going to release something that is going to damage the cell. So what happens then is we get the protective antibodies produced against the toxin so that their interaction is neutralizing the biologic effect. All right, and that is agglutination and precipitation, preventing harming of the recipient by those bacterial toxins, and they are chemically inactivated. We get indirect effects, and they will activate components of the innate inflammatory response. They can activate components of innate, such as complement and phagocytosis, and we saw that with IgG and IgM. They are able to activate complement, and then phagocytosis to be able to eat antigen. So that is that classic pathway that we talked about with complement. All right, let's look at natural killer cells. So we talked about these a little bit earlier. Just what you need to know is that they are present in the adaptive immune response, but also the inflammatory response. So they're here in both. And remember that they can kill something without being exposed to antigen. So that is high yield. All right, again, remember that the antigen presenting cells are highly specialized hunter cells that capture antigen and present it and display it to the lymphocytes of the adaptive immune response. So let's take a look at antigen processing a little bit closer. So we have intracellular and extracellular proteins that can be processed by specific pathways and are presented in association with either MHC class 1 or MHC class 2 molecules. Extracellular microorganisms penetrate the host defenses, endocytosed by the APCs, present fused to lysosomes, degraded by enzymes within the cell, and then presented. Intracellular, so there may be an internal antigen, because remember, viruses go into the cell and hijack the machinery. Cancer is related to cell proliferating and making neoplastic cells from one. We'll talk about that more in neoplasia. So something is within the internal environment of the cell. It's processed, presented on MHC class 1, activates a second signal, IL-2, and then we get cytotoxic T cell activation related to coming in and killing that cell then. This is just another synopsis, understanding that we have B cell receptors, and they look different, okay? So here's a T cell receptor, here's a B cell receptor. Synopsis of MHC class 1. Here is a look at class 1, okay? So there's something here. Virus, tumor antigen, we get proteasome, antigenic peptide, endoplasmic reticulum, and then to the top of the cell surface and presented as MHC class 1, ultimately getting CD8 cytotoxic T cells. When we talk about MHC class 2, we have an exogenous antigen brought in, phagocytosed, and then we have a couple different steps, but it's ultimately presented via the phagocytosis line, and then we have MHC class 2. All right, just to review macrophages, like I said earlier, we may be going over things multiple times related to where they're at in the innate versus adaptive. So macrophages are huge in the adaptive immunity. They are the big APCs. We have activation via 2 pathways, classical or alternative. Activation is a result of microbial products such as endotoxin. So we have endotoxin, and they activate the toll-like receptors and other sensors. Or we have the alternative pathway, which is induced by cytokines. Dendritic cells, most important for initiating T cell responses against antigen. They are located under the epithelium, best place to hunt microorganisms, right, after exposure from that primary and secondary boundaries that express high levels of major histocompatibility complexes. Antigen. So what is an antigen? Basically any molecule that is recognized and stimulates the cells of the immune system. It is recognized as non-self. All of that is not recognized as self. So that is considered an antigen. So when we talk about key response terms and recognition, these are the things that you must know what they are. So another thing we want to look at here is kind of the immune response as it's laid out for serology. So you will be doing testing. You need to know when the latent lag period is. So it's divided into two phases, primary and secondary response. This occurs in both the cytotoxic and the humoral. We have a latent or lag period of 5 to 7 days. This is when IgM antibody specific to antigen can be detected. IgG can be detected as well. IgG and IgM are about equal. This is referred to as a titer. If there's a high titer, it means we have more antibodies. The secondary response, this is a very rapid production of antibody. It's actually bigger than the primary response. And IgG is what is produced. Here is a look at that schematic. All right, so now we're going to look at alterations in immunity and inflammation. So first up, we're going to take a look at hypersensitivity. So this is an altered immunologic response to an antigen that results in disease or damage to the host. Two ways that this can happen is we get the source of the antigen of the immune system is attacking, or we can get a mechanism that causes disease. So just a couple definitions so you know the difference between things. So we have hypersensitivity is an altered immunologic response to an antigen resulting in disease and damage. Allergy is a deleterious effect of a normally benign environmental allergen that creates an altered body response. Alloimmunity is also termed isoimmunity, and this occurs when the immune system produces a reaction against tissues of another individual. Transfusions, transplanted tissue, or Rh factor with the fetus. The mechanism that initiates the onset of the hypersensitivity, whether it is allergy, autoimmune, or alloimmunity, is not completely understood, so we don't know what that is. Autoimmunity is the disturbance in tolerance of self. The immune system normally does not strongly recognize the individual's own antigens. Healthy individuals of all ages produce low quantities of antibodies against their own self, which are called autoantibodies, without the development of an autoimmune response. All right, so we do produce autoimmunity. But what happens with autoimmune diseases is this is when the actual immune system reacts to those self-antigens. And we'll get more into that when we actually define autoimmunity. Immunologic homeostasis is basically the steady state of tolerance, so steady state self-antigen, or lack of response to self-antigen. The individual's genetic makeup determines the degree of immune responses and the effects of those response. Okay, so let's take a look at hypersensitivity reactions. We have 4 distinct types of hypersensitivity, type 1 through 4. They are interrelated, meaning that these reactions sometimes happen together or sequentially. They can be secondary to disease or the primary cause tissue destruction, and they require sensitization. The mechanism are apparent in most hypersensitivity reactions. Type 1 is an antigen and specific IgE, which is directly attributed to those vasoactive mediators released by mast cells and basophils. Remember how I said that IgE and mast cells have high affinity for each other? This would be a type 1 sensitivity reaction. Most type 1 reactions occur against environmental triggers such as pollen, environmental allergies, food allergies, and then we have the bad one, which is anaphylaxis. This is part of the humoral because of IgE, right? So IgE, humoral, the body systems. So this is going to be B cell, and it is an immediate reaction. So in some individuals, we get exposure to an environmental antigen causes primary IgE production. That repeated exposures to the antigen is usually required to elicit IgE, so the person becomes sensitized to allergen combo. Relatively short lifespan because it rapidly binds to those Fc receptors on the mast cells. If further exposure of a sensitized individual to the antigen occurs, the one molecule of antigen can bind simultaneously to two molecules of Ig-Fc receptor complexes on the mast cells, activating internal signaling pathways and mast cell degranulation. And we get this IgE-mediated response. So that is what that looks like. So a type 2 hypersensitivity reaction. So this is a tissue-specific reaction. There are specific cell or tissues being the target of the immune response. So this has a lot to do with the HLA locus, which is human leukocyte antigen. And this has a lot to do with the major histocompatibility complexes as well. So these tissue-specific antigens, because they are expressed on the plasma membranes of only certain cells in specific tissues, only found on them and no other cells of the body. So these cells will then express that particular antigen. Other environmental antigens, such as drugs and their metabolites, may bind to the plasma membranes of specific cells and tissues and then target that reaction. We have 5 general mechanisms of type 2 reactions. The first one is IgG or IgM reacts with antigen on cell membrane surface. So then what happens is we can get destruction of the cell through activation of the complement cascade through the classical pathway, and we ultimately end up with that MAC complex and we get cell lysis. The second is the antibody may cause cell destruction by phagocytosis. They bind receptors on the macrophage. The phagocytosis of the target cell follows. The third is antibody and complement may attract neutrophils, and then they will chemotaxis for the neutrophils, deposition of complement. They bind the tissue of that Fc portion, and then the neutrophils release the granules onto the healthy tissue, and then we ultimately get damage to the tissue because of the neutrophil activation. The fourth mechanism is antibody-dependent cell-mediated cytotoxicity. This is a mechanism which involves the cytotoxic cells that are not antigen-specific, such as the natural killer cells, and they release toxic substances that destroy the target cell. The fifth mechanism is cell malfunction. It does not destroy the target cell, but rather causes it to not work correctly. In this mechanism, the antibody is usually directed against antigenic determinants associated with specific cell receptors. The antibody reacts with receptors and then modulates something within the cell to have it malfunction. Graves' disease would be what's going on here. And this kind of is just a picture of how all of that goes down. Type 3 hypersensitivity reaction. So this is an antigen immune complexes that are formed in the circulation and then they're deposited later in vessel walls or extravascular tissues. So this is also the humoral response because we have antibody, right? So anything with antibody is humoral. It is also an immediate response. So they may be deposited in tissues such as in celiac disease, lupus. So those IgG and IgM unite with soluble antigen, form that complex. That complex is then deposited in other tissues. One of the main ones here is Arthus reaction. So this is a prototypical example of a localized immune complex mediated inflammatory response. The immune complexes get deposited into blood vessels, joint, kidneys. You get fever, enlarged lymph nodes, rash, and pain at the sites of the inflammation. So here is an example of that. Last is type 4 hypersensitivity reaction. So this is the only delayed and cellular. So this is T site related, T lymphocyte. So this is reaction of sensitized T lymphocytes with antigen and do not involve antibody. So therefore it's cellular, not humoral, because there is no antibody. Lymphokine release, recruitment of macrophages, and lysosomal release. So type 4 mechanisms occur either cytotoxic T lymphocytes or lymphokine producing T helper 1 and T helper 17 T sites. Clinical examples here would be hypersensitivity reactions, graft rejection. They believe too that there may be a lot of type 4 components in autoimmune diseases, such as type 2 collagen deposition of the joints and rheumatoid arthritis. T cells against thyroid surface antigen contribute to the destruction of the thyroid. So here are T cells, which are what the cause of Hashimoto's thyroiditis is. Those would all be examples of type 4 hypersensitivity reactions. So here is an example of that. Alloimmunity occurs when an individual's immune system reacts against antigens on the tissues of other members of the same species. The most common would be transplant reaction or transfusion reaction. There are two clinically relevant examples of this reactivity. There are several neonatal diseases in which the maternal immune system becomes sensitized against antigens expressed by the fetus, and then transplant rejection and transfusion reactions. So transient neonatal alloimmunity, because the fetus is a hybrid between the mother and the father, it expresses paternal antigens that are not found in the mother, and that ultimately is what reacts. So we have mother plus father, and since the mother is the host, does not like dad, and we get a autoimmune alloantibody reaction. So strongest reactors, type A blood receives type B blood, reacts strongly because the circulating antigens result in agglutination or lysis of the cells. Type O lacks both, therefore the universal donor, and is going to not have a reaction. AB type universal recipients are also able to accept all antigens, and we will not have a reaction since there's mixing of AB blood. These harmful transactions can only be prevented by complete and careful ABO matching between donor and recipient. The RH reaction, so this is a group of antigens expressed only on the red blood cells. This group is most polymorphic, and that has at least 50 separate antigens, at least 5 major antigens, and a large number of rare variants have been identified. This is where the Rh negative mothers attack an Rh positive fetus, and it's basically a maternal fetal incompatibility. A disease called hemolytic disease of the newborn was the most commonly caused by IgG anti-D alloantibody produced by an Rh negative mother against the erythrocytes of her Rh positive fetus. Graft rejection, so this is the immune system attacks the transplanted organ or tissue. This is related to the human leukocyte antigens and the different MHC genetic loci. So there's a whole bunch of different loci that this would be genetically identified as. We have 3 types. We have hyperacute rejection, and chronic rejection. Hyperacute is immediate and rare, and that's when the circulation is reestablished to the graft area, and it becomes immediately white. It presents if there's a pre-existing antibody reaction to the HLA antigens. The acute rejection is primarily cell-mediated, and that's within days to months after transplantation, and that's basically the recipient develops an immune response against unmatched HLAs after transplantation. Chronic rejection is after a period of months or years of normal function. It's slow progressive organ failure, and this is from a weak cell-mediated months to years. But it's all related to these HLAs. So let's take a look at some immunodeficiencies. So disorders resulting from immune deficiency are the clinical sequela of impaired function of one or more components of immune and inflammatory response. So we have primary immunodeficiencies, which are a congenital issue caused by a genetic anomaly, whereas secondary is acquired immune deficiency, and it's caused by another illness. So secondary is always caused by another illness. Primary can be sporadic and not inherited. Up to 25% will have no family history, and that's called idiopathic. And it usually appears within the first 2 years of birth because we don't have the ability to fight off infection. Secondary, this may not be clinically relevant, all depends when it presents and what is involved. Can be very common, and some are very minor or severe, which would be HIV or AIDS. The hallmark of these disease processes is the tendency to develop unusual and frequent infections. So these are going to be these atypical frequent infections that really, everybody else in the family is healthy, but the child may present with some weird infections. You should always consider potential immune deficiencies if the individual has severe documented bouts of pneumonia, acute otitis media, sinusitis, bronchitis, septicemia, meningitis, and then also some of your opportunistic such as candida. All right, so we can get complement deficiencies, so they can be inherited or acquired. Again, infections are going to be the way that we present. Complements are pretty rare. They only represent about 5% of primary immunodeficiencies. We have a couple different types. We can have C1 inhibitory deficiency. So C1 is the beginning of all those complement pathways. So if we have an inhibitor deficiency, we may present with hereditary angioedema. If we have problems with C5 through C9 deficiency, you would see increased risk for such things as Neisseria infections, such as Neisseria, gonorrhea, or meningitis. If we have a C2 complement deficiency or a mannose binding lectin complement deficiency, we often do not see signs and symptoms that would be consistent with that. Now let's talk about the severe presentations of immunodeficiency. Severe combined immunodeficiency is the bubble baby. So those are patients that will not be able to be out in the world because they literally don't have cell-mediated or humoral immunity, and there's varying levels of where it's affected. There is multiple types of SID, and it all depends which ones are deactivated. The most severe form, and this is basically failure of the blood cells to develop. It's common stem cell of all white blood cells. Therefore, T cells, B cells, phagocytic cells never develop. They may have a few detectable lymphocytes in the circulation and secondary lymphoid tissues. The thymus is usually underdeveloped because of the absence of T cells. Immunoglobulin levels, especially IgM and IgA, are absent or reduced. There are at least 20 different forms of SID. So that's why just kind of understand this as an overview and understand that it's going to affect all T cells, B cells, also natural killer cells. You can have any combination of defects in them. So we have an adenosine deaminase deficiency, which helps eliminate waste products and buildup is toxic to lymphocytes. We also have cytokine receptor deficits. So we have decreases in cytokine signaling, which is necessary for the maturation of the B cells and the T cells. We would have MHC class 2 deficiency, and that's necessary for the activation of CD4 helper T cells, also cytokine production. So they will be highly susceptible to all processes such as fungal, viral, bacterial, protozoal infections. They cannot get vaccines. And bubble baby is the treatment, and they have to be sterile, isolated, and then potentially stem cell transplant would help them. Typical presentation: recurrent infections, failure to thrive, chronic diarrhea. You may find a diffuse erythematous rash. Absence of lymphoid tissue would be high yield, poor weight gain, and that comes back to that failure to thrive. So what are the risk factors of developing this? Athabascan speaking Native Americans, you can see the incidence here. Consanguinity, which is interbreeding, and then a family history of SID or an infant death. So if you would potentially have a family that had a previous baby that didn't make it out of that 2-year mark, that would be high yield, especially if your current patient is presenting with atypical infections. There is an increased incidence in the Amish and Mennonite and Athabascan Native Americans. The lesser form of this is called CVID, or common variable immunodeficiency. Again, it's going to have a typical presentation of recurrent chronic atypical infections. Same kind of presentation. You may not be severe enough, so you may give some live vaccines, and then you would see problems afterwards. So again, this is a hypogammaglobulinemia, so it's related to your gammaglobulins, which is the other word for immunoglobulins, and that is an X-linked genetic mutation resulting in B cell and T helper cell defects. This one usually presents in late childhood because it's less severe. This is the most common diagnosed immune deficiency. So there would be secondary causes of CVID. So anything that's going to impair production and loss of hemoglobins, myelomas, leukemias, medications, inflammatory bowel disease. You can see here 1 in 30,000, so it is kind of common, and it is the most clinically relevant immunodeficiency. The ratios of male to female are equal, and there are some rare forms that are X-linked. And then here are your risk factors: young child, early adult, family history, and protein losing state. Okay, so let's take a look at autoimmune disorders next. So autoimmunity hallmark is the immune system reacts against itself. Autoimmunity. There are alterations in tolerance. We're going to talk about immunological tolerance of self-antigens and the pathogenesis of that state. Genetically susceptible individuals are exposed to an initiating event. So with autoimmunity, we have a genetically susceptible individual and an environmental trigger. Both T and B cell responses are likely involved, and then there are certain pathogenic mechanisms that are common to all immune system diseases. Damage to self tissues mediated by the immune response. 1 to 2% of the US is affected, and it is more common in women of childbearing age. Classify that as 20 to 45, as high as 50, right? Because we still may not have menopause until after 50. They possibly believe it's related to estrogen, as this may reduce apoptosis of self-reactive B cells. Not entirely sure. This is an iatrogenic kind of situation, but they do believe, since it's more common in women of childbearing age, that it is related to estrogen. There is that environmental trigger with genetically susceptible individuals. There is an increased twin incidence, which gives us that genetic component. Most disease courses have relapses, remissions, and flares. So self-tolerance, as those lymphocyte receptor sequences are generated completely at random, remember that it is DNA controlled, we have that region on the T cell receptors and the B cell receptors that is generated so that we can identify antigen. Well, we also have with self. So we have self-antigen at this time. It is completely at random. So this randomness results in many diversified receptors that will recognize nearly any antigen the immune system encounters, right? That's what we want it to do, as we talked about it with the B cell and T cell clonal selection. One of the ways that the immune system checks these receptors is known as self-tolerance. This is used to reduce the scope of the lymphocytes' recognition and subsequent attack on foreign antigens. Some of these self-reactive cells escape screening and immigrate into the periphery. So these autoreactive cells go into the circulation and result in catastrophic immune attacking against the individual's own tissues. One stage of the immune tolerance occurs at the deletion of the autoreactive B cells in the bone marrow, which is referred to as central tolerance. So we have 2 steps in this. Here is that central tolerance. So we have the pro-B cell, which is in the bone marrow. So that's this nice little diagram. Maturation of the B cell, we get that chain arrangement. So we get those immune globulins. And then what happens is we get that immature B cell, and we get this receptor editing, making sure that it is a good tissue. This is when there's a large number of autoreactive B cells. And if this is not a good cell, we basically get apoptosis, and that is clonal deletion if it is exposed to self-antigen. Some immature B cells escape this receptor editing and clonal selection and determination here, and they will then escape into the peripheral circulation. And usually we have another way of identifying these receptors, and we will try to catch it again in peripheral tolerance. For autoimmune diseases to proliferate, we have to have a defect in both peripheral and central tolerance, and it allows that bad B cell to get out of the area. So in this process of tolerance, immature self-reactive T and B lymphocyte clones are recognized self-antigens during their maturation in the central, which is the primary lymphoid organ, such as the thymus for the T cells and the bone marrow for the B cells. They are killed or rendered harmless. In developing lymphocytes, random somatic antigen receptor gene rearrangements generate diverse antigen receptors, many of which by chance have a high affinity for self-antigen. These mechanisms of central tolerance eliminate these potentially dangerous lymphocytes. Okay, so the body has a way of kind of detecting self-antigen and trying to get rid of it, and that's what this process is. So central tolerance is in the bone marrow for B cells and in the thymus for T cells. When there are immature T cells expressing T cell receptors specific for self-antigen, these antigens in the thymus signals are produced that result in the killing of the cells by apoptosis. The body is trying to get rid of these dangerous cells because what is ultimately going to lead to is an autoimmune disease, which is what we want to avoid. It is not imperfect. Not all self-antigens may present in the thymus or the bone marrow. Some of them may present when we get into the periphery, and then that is when they would be detected by the peripheral tolerance. This is called negative selection if they get out of the central tolerance, meaning the body did not recognize it. Then we have several mechanisms in the periphery, such as anergy, and that is basically when lymphocytes recognize self-antigens that may be rendered functionally unresponsive, which is this anergy. So remember we talked about co-stimulation. So a lot of these T cells that have these associated molecules will not bind. They don't get that secondary stimulation. And then we also have suppression of T regulatory cells. And remember that the T regulatory cells prevent immune reactions against self. So if they're lacking, then we would have an expression of autoimmunity. Another way is deletion of apoptosis. So normally T cells recognize self-antigens, may receive signals that promote their death. By apoptosis, depletion of these T cells can occur in the thymus and also in the periphery. And again, there's a lot of steps involved in this, but what you must understand is that we basically recognize self-antigen, but our checks and balances system does not get rid of it. So we have an induction of autoimmunity, because these defunct self-antigens get into the system, get out of the bone marrow, get out of the thymus, into the periphery, don't get picked up by the peripheral tolerance either, and basically get into the blood system and get deposited in other tissues. So what we mean is loss of tolerance when we're talking about autoimmune diseases. One of the biggest ones is lupus. So this is antigen-driven immune-mediated disease of IgG antibodies against double-stranded DNA and nuclear proteins. We get anti-nuclear antibodies. This is classified as a type 3 hypersensitivity reaction, as we get deposition of those immune complexes and binding of various antibodies to various cells and tissues. So remember that this is the ones that go to other tissues and deposit themselves. It is fairly common and has a prevalence as high as 1 in 2,500, predominantly women of childbearing age with a female ratio of 9:1, so it's huge. So etiology is unknown, so this would be idiopathic because we do not know exactly why lupus starts. So what theories they have are those hormonal factors like we talked about with the estrogen. Certain drugs can elicit this autoimmune response. There can be infectious, non-infectious viruses such as the Epstein-Barr virus. So here's that epidemiology. So women of childbearing age, 9:1 or 12:1, depending on what source you look at, is the female to male ratio. You can see after we get with lower estrogen levels, we're getting towards menopause. Look at the change in the ratio, 2:1. It does affect African descent more than Asian and then white. The severity of the disease increases in African and Asian descent. So that's high yield. So when we look at the patho, we have those genetic factors, genetic contributions, the loss of tolerance. So basically loss of tolerance to self-antigen, B cell pool by several mechanisms, and then also regulation of the T helper cells and T regulator cells. And that's all goes back to this entire process. So regardless of what autoimmune disease we're talking about, it is related to these self-reactive clones getting out in and depositing in other tissues. Your signs and symptoms are dependent upon where the complexes are deposited. We can get rashes, and they commonly occur after sunlight exposure and more photosensitive. The discoid rash, so this is an erythematous raised patches with keratic scaling, so they kind of look almost like psoriatic. Overwhelming fatigue, weight loss, fevers, they can have oral ulcerations, alopecia. The way that you're going to work this up is CBC, we're going to be looking for anemia. There can be some issues with also some cytopenias because of where autoantibodies, such as the red blood cells, white blood cells, platelets, they are opsonized and promote phagocytosis and destruction. That's how we get the cytopenias. We may have this antiphospholipid antibody syndrome, and they may develop the venous and arterial thrombosis, and they may have spontaneous miscarriages, cerebral ischemia, so that's why you would be checking your prothrombin time. You need to look at your kidney function, electrolytes, because this can deposit within the kidney and cause some glomerular nephritis secondary to lupus. You would be looking at inflammatory markers such as the ESR or C-reactive. They are non-specific, but it would go to that inflammatory response. Here are all the hallmark high yield, so ANA, which is that anti-nuclear antibodies, the double-stranded DNA, and the Smith antigen. These are all pathonomic for lupus presentation. You want to check your cardiac profile as well. There's the butterfly rash, and then this is the discoid rash. Okay, so that is the immune, adaptive, and autoimmune lecture.